Good afternoon, everybody. Welcome to today's Jewish Gen Talk with Dr. Lawrence Fagan. My name is Avram Grohl. We will get started in just about a minute. We will let people log in. And as we always ask, please post in the chat where you are logging in from. We have people who have registered from all around the world. We'd love to know where you're from. And we'll get started in just about a minute. Barry Finkel is first up on our list from Chicago, JGS of Illinois, M. Larkin in Medford, Massachusetts. Leslie Wacht from Arcadia, Wisconsin. Looking forward to seeing you next week, Leslie. Phyllis Radek from Las Vegas. We have Sabine, I think, from Rehovot in Israel. I'm so glad that you're with us. Somebody from England, Phoenix. Madeline Eisenberg in Los Angeles. So glad you're with us. Ruth Weisgarth went quickly, I think, from Israel. Thank you for being us. We have someone from Seattle, Milwaukee, Wisconsin. Eric Block from Milwaukee, Wisconsin. Rose Liebler from Jerusalem. Alona Abinezer from Jerusalem. Sarah Weil from Jerusalem. Sinai Zilper from Kiryat Ono in Israel. Linda Johnson from Washington, D.C. Carol Skolnick from Columbus, Ohio. We have Lori Matola from Teaneck, New Jersey. Len Levine from Bethesda in Maryland. Welcome, everybody. We're going to get started in about 30 seconds with our talk for today. And you can let us know in the chat where you are logging in from. We have someone from Finland. Welcome. We have Bernard Solomon from Monroe Township in New Jersey. Mike S. from D.C. We have John Steckel from Vancouver, Michael Scholar from Houston, Texas, Glenn Ginsberg from Denver, Colorado, Rosalie Fox from Ottawa, Canada. We have Sylvia Finzi from London, United Kingdom, Cheryl from Los Angeles, Anne Schumann from Monroe Township in New Jersey. We have Rita Scher from Johannesburg, Tennessee, if I'm pronouncing that. We have Yoni from Israel, Gail Jaffe from Los Angeles, David Robinson from Winnipeg, and Hillary Wolf from Los Angeles. Thank you so much to everybody who's joining us today. My name is Avram Grohl, and I oversee Jewish Gen, and today's talk is part of our ongoing Jewish Gen Talk series of educational webinars. And today we will focus on genetic genealogy. Our speaker today is Dr. Lawrence Fagan, who, if you are involved in our Jewish Gen educational efforts, needs no introduction, but his career began in the early days of computers and applied to medicine and biology. He held teaching and research roles as this field developed over many years. And during the last decade, he became interested in the field of traditional genealogy and especially genetic genealogy. He has met many relatives through these efforts, and for the last 4 years, he has taught the Jewish Gen introductory and intermediate genetic genealogy courses. You can, of course, learn more about his courses and all of our offerings at JewishGen.org/education. And we really feel privileged that he is joining us today. A few notes before we begin. The session is being recorded. We will make it available to all of you, our registrants, via the Jewish Gen YouTube channel. And a link will be sent to you either later today or tomorrow with a link to the video along with a link to a survey that we hope you will complete. And also just note that since you registered and since you're participating, you will be getting access to the video in advance of when it's made public. If you'd like to ask any questions, Dr. Fagan is happy to answer them. Please post them in the Q&A section, not in the chat. It will be very difficult for us to go through the chat for the questions. And finally, just a reminder that if you're not subscribed, please sign up for the Jewish Gen discussion group. That is a great place to connect with others, to ask questions, to share advice, and to stay informed of what's happening throughout the Jewish genealogical community, including having webinars like this. So we are really so glad that you're here today. And without further ado, I pass it over to Dr. Lawrence Fagan.
Hi, my name's Larry Fagan, and I'm currently living in a rural area outside of Spokane, Washington, in eastern Washington. As Abraham mentioned, I've been working in this area for about 12 years after a long career in computers and medicine. So let me share the screen and we'll get started. So the purpose of today's lecture is really to give an introduction to genetic genealogy. And by that I mean doing DNA testing to verify relationships in your family tree and to locate unknown members of your family tree. There are other uses we'll get to later, but those are what we're going to concentrate on. But before I go into a lot of detail, I want to introduce you to Boris G. I met Boris through genetic genealogy applied at 23andMe. And Boris told an amazing story. His mother was one of 6 siblings from Belarus, what's now Belarus. 4 of those siblings and their families came to the US in 1900 to 1910, which is a very common pattern. But 2 families could not leave Eastern Europe at that time. They stayed in touch until 1937, when they were no longer able to communicate. Boris did not come to the United States until 1995, when he was able to leave Russia. He had no idea where those other 4 families were located. He talked to his family, and his grandson decided that they were going to test and put his data on 23andMe. About the same time, I had retired and I put my data on 23andMe, and the 3rd person on the list of closest matches was someone I had never heard of, from Boris. And it turns out that I was from one of those families that came over to the United States. And he was in close touch with the 2 families that stayed in Europe and were not able to come over until 1995. And what he said was, I've been looking for those other 4 families, and this was the way to connect. His surname was not one that I knew, but through the kind of techniques that we're talking about today, we were able to connect, we were able to meet. I was able to meet other parts of the family. This is what genetic genealogy is all about. So I dedicate this to Boris. Now, in the ideal world, the idea would be you do the test, there's a database of everyone who's tested, and a family tree pops up with all the answers that you couldn't get with your paper records. Unfortunately, it doesn't work that way. The problem is that it's a lot more complicated, and here are some of the complications. The first is, in the US, we have 4 main testing companies, so there are 4 different databases of people to match against. There's not one type of genetic test, there's actually 3 different kinds of genetic tests. And so you have to know which kind of genetic test gives you the answers to the kind of questions that you're asking. And in addition to those 4 websites having the ability to manipulate and match you to unknown people, there are also many other programs that exist. For example, one we'll show you today is DNA Painter, which gives us some very useful information. But there are also programs like the Auto Cluster program that let you match all your matches against all your other matches to try to see if they can group them into people who may be related to each other. Well, this is a table developed by Alice Ramsey of relationships. Here I am. You can see parent, grandparent, great-grandparent. You can see sibling, first cousin, second cousin. At the end of the day, when we finally figured it out, it turns out that Boris, who shares almost 5% of his DNA with me, was my father's mother's mother's sister's son. So that's the relationship, first cousin twice removed. But let's talk about how we find a person who's not in your family tree, figure out how they're related to you, and how to put them into the family tree. That's the purpose of this effort. So first of all, we're going to use one of those websites that's available in most places called MyHeritage. We're going to use one of the three tests. We're going to use what's called the autosomal test. And we're going to try to find out how are we potentially related. Now, each of these websites will give you their best guess. And MyHeritage best guess was that Boris was my first cousin's grandson. I'm pretty sure that's not right because when I met him, Boris was 95 and I was 65. So this isn't going to work. So there's some practical things you have to take into account. But the other thing MyHeritage said is that we shared 4.6% of our DNA. And that is a lot. My first cousins that I've also tested shared between 8 and about 11%. So Boris is about half as much DNA as my first cousins. So definitely a close relative. So how can we find out what's the real possible relationships? So one of the websites that I use and many people in this field use is called DNA Painter. And a part of DNA Painter is called the Shared Centimorgan Project. So centimorgan is a measurement, a rough measurement of how much DNA you have. It's just a number. But the 4.6% that MyHeritage mentions corresponds to about 340 of these shared centimorgans. So what Blaine Bettinger did several years ago was he got people who had known relatives and he found out how much you matched, and he made tables that said if you match between this length and this length, then here's the most likely relative. This is the relationship that you have. And then Johnny Pearl, who's also involved in creating this DNA Painter site, then created a digital tool that you can go on a website and you type in either the number of centimorgans or the total percentage. And what it does is it says, here are the probable relationships that you have. So for example, DNA Painter, this particular tool says I have a 50% chance if Boris and I share 4.6%, that could be a great great aunt, could be a half first cousin, could be a half nephew. Actually, great great aunt doesn't sound like the right relationship, but great great uncle is a possibility. We have a 45% possibility that that he is a half great great nephew, second cousin, or what it turned out to be, first cousin twice removed. There are some other possibilities, but you'll see that him being a great uncle or a great nephew is just not possible. So we're trying to understand what are the possibilities. The other thing that shows up at this website is the ability to create a graph. So this is a table like the one I showed you at the beginning, which shows myself here, child, my grandchildren, first cousins, second cousin, third cousins, and it tells the range of centimorgans that would be expected. Now, a lot of these are grayed out because these people would require more shared DNA than the percentage that I have with Boris. These people are grayed out because they typically share even less DNA than I share with Boris. But those in the middle are the possibilities, the same ones we saw in that chart, that show where it's likely to be relationship that I share with Boris. And I'm here. Boris is over here as a first cousin twice removed, which would include this number of centimorgans. So this kind of tells me how far away he is from me on the tree, but it doesn't answer the question of where is he on the tree, because we want to know that too. So we'll get to that in just a moment. But I'm going to go just a little bit in the biology, not deep into the biology, but I wanted to remind you that in the nucleus of most of your cells is the DNA that we use for this testing. Fortunately, you get it by a cheek swab or by a sample of sputum. But what they do is break down the cells and they get to the DNA. And what you see in everybody's DNA is 22 pairs of chromosomes that all have different information on them that helps make your body function. And then males have an X and a Y chromosome, and females have an X and an X chromosome. So you have one or the other. Each of these has functions, although we're not going to talk about the health functions very much here. But one of the reasons that I picked chromosome number 6 is that is where the immunological pattern of your body is stored. So, for example, if they're looking for bone marrow transplants, one of the things they do is look at a particular little section of chromosome 6 to see if you are a match. So this matching process is also used in a medical setting as well as for our work in genetic genealogy. So I'm going to zoom in on chromosome 6. And one of the things that is very important to understand is that these pairs of chromosomes are come in to the chance of when you're making a child have to be managed in a certain way so that you don't run into a problem. And the problem is that the father has a complete set of these 2 chromosomes. The mother has a complete set of 2 chromosomes. And the child has a complete set of 2 chromosomes. So if we take all the genetic information from the mother and father and put it into the child, it won't fit. What we have to do is take half of the information from the father and fit it into this chromosome, and half of the information from the mother and put it into this chromosome. So what we're going to do is this chromosome came from the father's father. This chromosome came from the father's mother. This chromosome came from the mother's father. And this chromosome came from the mother's mother. And so what we're going to do is these 2 are going to be copied, segments are going to be copied to build up the child's paternal chromosome and the child's maternal chromosome. So imagine that you go and take a segment of here and copy it, and then you copy a segment of here from the maternal side of the father, and then you go back to the father's side and copy some of that. So this chromosome here is all coming from the father, but the problem is it's coming a little bit from one of the father's parents and a little bit from the other of the father's parents. Likewise, you do that here. So we copy a little bit from this chromosome from the mother's father, and we copy a little bit from the mother's mother. And so that's how we make up these chromosomes. That means 50% of the DNA is lost in each generation. And so after 5 or 6 generations, this kind of autosomal DNA testing is very hard to find connections because data gets lost at each generation. So on the right-hand side here, let me show you a graph, a graphical way of looking at this. Let's assume that the father's components are made up of this dark blue for the father's father and the light blue for the father's mother and dark red for the mother's father and light red for the mother's mother. And when we go through this process to make the child, we get a little bit of dark blue from the father's father, then light blue from the father's mother, then dark blue again. And over here, maybe we get the light red from the mother's mother, and then we get the dark red from the mother's father. So that's how this information is condensed into this kind of situation. Now we're going to go one more generation. So what I just showed you was this blue square here. And imagine now that this is a mother and father. They have 3 children, and these are the grandparents up here that have those solid bars. So the same thing's going to happen with the mother's family. The mother's family is going to have some of the dark green and light green and on one chromosome, and the orange and the yellow on another. When we go to the next generation and we take these from the father and these from the mother, we're going to get 2 chromosomes, but they're going to be mixed up. So this is going to be the blue and the red, and then we're going to have the yellow and the orange and the green, and similarly for the other 2 siblings. You'll see that they all, unless they're identical twins, you don't get the same mixture, and that's why siblings don't have the same appearance, even though they're coming from the same parents. So this is the process roughly that's going on behind the scenes. But the key thing that we want to do when we're doing genetic genealogy is we want to look not where they disagree, but where they agree. So you'll see that there is a common segment in all 3 grandchildren that are coming from the paternal great grandmother, which is the light blue. And you'll see that it's in about the same place in each of these 3 images. So this is a match that these 3 siblings have in common. Now, this one kind of extends over, but that doesn't match over here, but it does match to this person. And so when we're doing a 3-way match like this, we want the part that everybody has. And that's what's going on here. So in the process of looking at this autosomal DNA, we see that the DNA is cut in half at each generation. So from each of the original information that we had about the parents of this person, there's only going to be small bits of it, about a quarter left in each of these grandchildren down here. We're going to concentrate on the segments that match between the individuals. And the closeness of the relationship is often related to the overall number of shared segments and the length of the longest segment. That's why my match to Boris was so meaningful, because 4% is a very large match. And so you learn the ranges that are meaningful as you do more work in genetic genealogy. So all of the programs except for Ancestry, unfortunately, have what's called a chromosome browser, where you see chromosomes 1 to 22, those autosomal chromosomes. And what these places shared in purple are the parts where Boris and I match. And in particular, I was talking about chromosome 6, so we can see a little bit at the end, little in the middle, a little bit near the other end. The other thing that this program is telling us is that the largest segment is 38 of these centimorgan measurements, and we have 18 shared segments larger than some cutoff. And that together we share 4.6%, which corresponds overall to 326 centimorgans. But there's a problem with this matching process. In fact, everything I've told you assumes that the families are unrelated to each other, that the bride and the groom are not related. But the problem is, because many, particularly in the Ashkenazi families, were in small towns that were founded by very few people, and often there were marriages where cousins married other cousins. And so this assumption does not hold true. And so what happens is that instead of these colors not matching until the progeny of the husband and wife, uh, there's going to be blue on this side of the family, and there's going to be green on this side of the family. And so these measurements here are not as clear cut. Now, Dr. Leah Larkin from the DNAgeek.com put together this family tree. This is actually French Canadian, but they have exactly the same kind of problems that the Ashkenazi Jewish families have with regard to people marrying within the families. And so this is not from genetic genealogy, but from traditional genealogy. And they've gone all the way back to the 1600s, where there were very few individuals that created the community in the first place. And so many of these people are related to other people in their family tree in multiple relations. And this was plotted using another special software called learnforeverlearn.com. So let's zoom in on just one little corner. I think it's this corner right here, and see what the effect is of this process, which is called endogamy. Okay, so here's a little close-up of part of that. And we're interested in this individual down here, who's marked with a yellow marker. Assuming that blue stands for male line and pink for female line, that this person's father, grandfather, the parents, so the paternal grandfather's parents, their parents, are these two people over here. And in this true family tree, those same two people are the mother's mother, maternal grandmother. Their parents are the same. So that means that this person down here is going to have twice the contribution from this family, because there are less great grandparents than you would expect, because they went down two different lines and then those people married and had a child. So that genetic information is doubled. So what are the effects of endogamy, and what are we going to do about it? Well, the first thing is that distant relatives may appear as closer relatives because you related to them in multiple ways. It's kind of like that rearview mirror that says things are closer than they really are. And so, for example, and this happened in my family, we had someone that claimed to be a second cousin relationship. but we were able to trace that it was actually a fourth cousin through both a father and a mother, just like was in that picture. So these add, so two fourth cousins looks as close as a second cousin, but they're really much further away from you on the tree. Secondly, because many of these communities had the same few individuals that formed the communities, the whole community may have DNA that has segments that match. And Ancestry calls these pile-up regions. And Ancestry has, for example, software that says we can find out with our community of the millions of people who've tested where we have this situation where we're looking way back in history to distant relatives. When we find those pile-up regions, then we know that that is looking way back in time at old ancestors and not at recent family. And so they actually subtract out those sections. Other programs don't do that, and so you have to adjust in different ways. One of the ways that people adjust is we're not even going to call it a close match unless we have a larger amount of DNA. I want to see, say, 20 centimorgans as your largest match, or 25 centimorgans, and I want to see two more segments that are at least 10 centimorgans. So there are all these rules that are mentioned about how we're going to say, take into account the fact that, particularly in the Ashkenazi family, that there's a lot of endogamy. But as I said, this same issue shows up in French Canadians on a Greek island when explorers came over and there were a small number of individuals that started the community. So not only do we want to know how far away someone is, it's nice to know that Boris is my first cousin twice removed. But the question is, how do you place them into the family tree? Because that's really what you're trying to do. You're taking your traditional trees that you've built and then adding in these individuals that we didn't know or we're not sure where they are um, into your family tree. But it's a very interesting process. Is if I take my DNA and match it to someone like Boris, that doesn't tell me anything about whether he's on my mother's side of the family tree or my father's side of the family tree. We just know that we match. We actually need to bring in another person if we're going to use the genetic approach. Now, many of you know the traditional approaches, for example, surnames. We could try to match surnames. Boris's surname didn't match anyone I knew in the tree. We could try to look at where they came from. Fortunately, my mother's from the Ukraine, what is now the Ukraine, and my father's family was from Belarus, which is where Boris was coming from. So that kind of would have told me father's side, but it's better to see if there's another approach. And the other approach is to use other individuals, kind of sentinel individuals that tell us more information. So we could use siblings, but that's this have the siblings are going to have the same father and mother, so that's not going to tell us anything about which side it is. And we could take my father's sister, who was led to my first cousin, and so if it's someone that I match, and my first cousin match, and my first cousin does match Boris, then that tells me it has to be related to these two grandparents, my paternal grandfather or my paternal grandmother, because as a first cousin, we share these grandparents and it's not related to these grandparents. But if I want to distinguish between paternal grandfather and paternal grandmother, then I actually need to get someone who's only on one side of this tree or the other. So I need a second cousin, say the sibling the grandchild of a sibling of my paternal grandfather. If I have that person matching me and matching Boris, then that tells me that they're on this part of the tree. So having cousins that you know where they are in the tree help you figure out where the person fits, even if you've never tested these people, because these people may have passed away, but they're too old to test. They don't want to test, but you can use the second, and if you go with third cousins, you can go up even another generation. So what we do is three-way testing to kind of pinpoint where in the family tree someone might be. So this process is called using shared matches. So typically you match with yourself, with someone you know, because you know where they are in the tree, and this unknown match, like Boris was originally, to kind of figure out which corner of the tree is this person most likely to be in. So the more relatives you know on a site, the better you are able to place these unknown matches. Now, shared matches is very powerful, especially if you can figure out how close the matches is. And something that's very interesting is Ancestry turned on the ability to do this shared matches work in the last few weeks. But unfortunately, they charge a fee for extra services that allows you access to this. Another thing that comes out of using this shared match approach is that you may want to encourage family members to test to provide for more known matches that you can use as a guidepost to the individual that is unknown that you're trying to place in the tree. And this, of course, is very complicated, made more complicated by the fact that endogamy may be going on. And so people are matching in multiple ways through different sides of the family. There are certainly people on my family that match between my father's side and my mother's side, which confuses this process. So we've been talking entirely about one kind of test, the autosomal test. And it uses those 22 pairs of chromosomes that we saw in that graphic. Because information, about half of the genetic information is lost at each generation, you can only go back about 5 or 6 generations. And because of that process I described, where little bits get copied and different people get different bits of DNA inherited from their parents, it can be a situation where you actually have a relative, like a third cousin, but they don't match. So 10% of true third cousins won't match, even though that they are real true cousins that exist. And you may find that if you go and use if you use other relatives like a sibling or your first cousin, that you can actually make the match to that third cousin from other people, so that it is a true match. But the problem is that too little of the DNA was left to make the comparison between you and that person. There's another kind of testing that uses the Y chromosome. And this can look back thousands of years along the paternal line. It's particularly useful if you want to look back and find out which surnames people adopted in the 1800s, when in the Pale of Settlement, many were required to adopt a surname rather than person, son of this person, or daughter of this person. And so as those surnames were adopted and people made up names based on where they lived or other non related items, you can see the surnames of other people that match along that Y DNA. Likewise, we can use the mtDNA, and this uses mitochondrial DNA, which is located in the cell, but outside of the nucleus. And this allows us to look back thousands of years along the maternal line. So your mother's mother's mother had this same mitochondrial DNA. It does have some mutations, but it happens very slowly. Uh, and there are, for example, 4 groupings that are very commonly associated with Jewish ancestry. So there's a lot more detail about the Y-DNA and the mtDNA than I can go in this introductory course lecture, but I want you to realize that there is other tests that can look at certain specific things, and it really depends on what questions you're asking genealogically that can determine which of these tests you should use. But the main one is really this autosomal test. So one strategy that many people in the field encourage is to start with the ancestry autosomal test. It's got the largest number of testers to match against. Then you can move that data without having to test again to MyHeritage and to FTDNA, although there are some fees associated with being able to search once you've moved the data. So you don't have to test multiple times in order to get your data into 3 of those large databases. But if you need more information, more matches, then you should do the 23andMe autosomal test. The reason you have to do this test separately is that both Ancestry and 23andMe do not allow you to move your DNA data to those sites. You can move it out of those sites, but you can't move it into those sites. So that's why it makes sense to start at Ancestry, move it out, and then potentially test it 23andMe. If you are particularly interested in your father's 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 line, you don't know much about that, then the Y testing might be useful. If you want to look at the mtDNA result and look at the mother's 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 line, then that can be useful. Now, Boris and I don't match on mtDNA because my mother was from a entirely different line than his mother. But my first cousin is her mother's 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 mother is Boris's mother's mother, and so has this same mtDNA. So you can do that kind of matching to verify the result from the autosomal testing. So let's kind of recap some of the pros and cons of genetic genealogy. You can find new family members. You're following the biological rather than the paper relationships. You can explore ancestors along paternal and maternal lines going back thousands of years. You can also look at the ethnicity and compare that across generations. But the problem is there are some significant negatives as well, and there are really serious ethical questions. The data that you've created can be used by third parties. So for example, 23andMe sells the data to drug companies. They ask your permission, but the way the drug companies do it is they ask you what diseases you have, and they find which bits of DNA are showing up in the people who have those those diseases. So they can use this to develop diagnostic tests, for example, which is a good thing, but it's your data, and that data can get out if there is a problem. There's also the use of this data in criminal and forensic investigations, either with DNA left at a crime scene, or if they have a forensic situation with a body they're trying to identify. Unfortunately, some of the early work in this area was done without telling people that they were using the DNA. Basically, they took the crime scene data and were able to basically make a kit that looks like the kit that you would take from scraping your cheek. and then put in the databases. Now people can opt in if they want to be used for criminal or forensic investigations, and they're only at certain sites that are doing this, but it is a potential issue. In the United States, we have a law saying genetic information can't be used against you for insurance purposes, but because if someone had access to your genetic data, they could know, well, you have this risk of, say, Parkinson's disease, or you have an increased of breast cancer. And then there are the effects on the family if you find what's called a non-parental event, or NPE, and find out the person you thought was the father or the uncle was not that member. And it's, you read stories every day about they did one of these tests, they found a relative, they established a family, but there are also plenty of stories about people where they found this out and it caused significant psychological problems in the family. And the worst and most recent situation was a break-in that happened in 23andMe. Because of the tools that are at 23andMe, if they broke someone's password, they also could get the names of all the individuals that match those individuals. And they could potentially sort those by ethnicity and create lists of people with certain ethnicities, which itself is pretty scary. So there are a lot of pros and there are a lot of cons. Overall, I'm still finding it extremely useful to do this kind of research, but I'm very aware of this. And I notice that when I look at 23andMe now versus 23andMe at an earlier time, that some people have dropped out, partly because they are required to sign in and verify again, and some people may not be available to do that. So this is just a little corner of genetic genealogy, and there's a lot more information out there. First of all, I introduced Blaine Bettinger, who did that shared DNA project. He has written a book called The Family Tree Guide to DNA Testing and Genetic Genealogy, which is very good and is available on Amazon and other locations. Many of these people have blogs. Blaine has one called the geneticgenealogist.com. Kitty Cooper has one called blog.kittycooper.com. And Dr. Leah Larkin, who provided that French-Canadian example, has the DNAgeek.com. There are Facebook groups. There's a Facebook group for each one of these companies. There's the regular genealogy Facebook groups, but there's also a specialized group for Jewish DNA for genetic genealogy and family research. So I encourage you, there's lots of good people there to take a look at that website. And I hope you might be interested in learning more about genetic genealogy from JewishGen. I teach 2 classes which are coming up shortly. A very introduction to genetic genealogy. You don't have to have tested in order to do the introduction. That's coming up starting next week and runs about a week. And then a more in-depth course, kind of at the intermediate level of genetic genealogy, which runs from the end of August to near the end of September, where we take your results and use a number of the tools I mentioned and more to get an idea of how you can answer your questions and build your family tree. So I'm going to stop sharing, and I'd like to open it up to questions. And Rami?
Thank you so much. That was really so informative. We do have some questions and they are in the Q&A.
Okay, and I see those. The first question comes from Jill. Says, I'm hoping you will clarify what the DNA numbers are when 2 relatives marry. My parental grandparents are possibly first cousins. Yeah, so one of the things that they did um, uh, is an extension of the shared DNA project that I was mentioning, where they asked people from endogamous populations, particularly Jewish, Ashkenazi Jewish individuals, to measure their relatives and what they saw. And so there's another table which takes into account general endogamy. When you have a close relationship like this, then that definitely uh, is going to make people much closer, and so it makes it much harder to tease out. I don't think that the table that they've done will answer this question specifically. You get the same kind of problem if you have, say, two identical twins marrying two identical twins. Then even though the child is typically a niece or nephew, they look like siblings. So you're kind of halfway to that situation. Okay, so the next question is it possible with one Jewish parent to not show any Jewish genetics? Both parents have Eastern European ancestry. So this is complicated. First of all, the ethnicity part of of these programs is one of the more speculative parts. The autosomal DNA I trust to a certain extent, but the other part is more difficult. The way that they do the ethnicity is they get what they think are the populations, and then they look and see which segments at the DNA level are common between these individuals. But the problem is a lot of these um, populations have overlapped over time. And so it's hard to kind of get a very specific answer. Uh, the way I can answer this question is when we see something in most of these programs, like 25%, Eastern European Jewish, then that usually means there's one grandparent that is Jewish. But it doesn't mean that it will catch all the situations. So it's possible. Uh, I think if you are seeing percentages like 1% or 2% of any ethnicity, I would take that with a grain of salt. If you're in the 25% or 50%, then then that's a different story. But it is more complicated because there was intermarrying with populations that Eastern Europeans may have married non-Jewish. There people uh, uh, can convert, which is not a genetic thing, and so it's pretty complicated. I do not have a handout. So what is the difference between 23andMe and Ancestry is asked by Bruce. There are two different websites. One thing that's nice about Ancestry is they have your family trees from your traditional genetic genealogy. And so with some of the advanced tools, they can look and see if you have common ancestors by looking at those trees. Ancestry has the most people who've tested, but 23andMe has the chromosome browser. So each one's a little different. MyHeritage has some very nice tools that let you compare the DNA and see if you match exactly DNA per DNA base pair. And so each one's a little different. If you get involved in this at a deep level, you will find that you probably end up being in all of these databases. And there's another one called GEDmatch, which doesn't allow testing, but which allows you to move it from any of these sites to that common site. And so you can test someone, might be able to see someone who tested at MyHeritage or 23andMe without having to actually move your DNA to those other sites like MyHeritage or 23andMe by testing at GEDmatch as kind of a common area. Um, uh, so someone asked, when was Boris born? So he was 95 when I met him in 2013. So probably about I can't do the math quite quickly, but in the first decade of the 1900s. So um, he was 30 years older than me, but we are two generations apart. And that's the problem, particularly if you had multiple family members over time, you can have it so you have a generation apart, but the ages are not that different. Julia asks, um, I've heard that identical twins have different fingerprints. How can that be if they 100% match DNA? So I am not an expert in identical twins. I know are small differences and there are biochemical small differences, but for the purpose of um, looking at them with the genetic testing, that like we're talking here, for matches, they're going to look and have they will have had that same pattern on the wide scale. The next person is asking, I'm trying to find my biological father's, and endogamy has created many brick walls. Can you provide an example or best practice for how to identify an unknown biological father if you don't understand how your thousands of matches are related? So first of all, there are some groups on Facebook that specifically are here to solve those kind of problems. Unknown parents where you're trying to find it using genetic techniques. The second thing is there are individuals which do this as a business um, that are a way to answer that question. And I think that that's an important uh, it to answer this kind of difficult question, you need to go to people who do this as an expert. Uh, my sister, biological sister, had her DNA tested. Is there any point in having mine tested? So the answer is yes. Even though you will match very similarly, you're going to have a different mix of DNA that was copied from your parents than your sister has. And so it will allow you to identify more people because of that information. For example, I mentioned that 10% of third cousins may not match because the DNA just wasn't in the same area. But but by testing you as well as your sister, you might be able to find the match there. So I think it is worth doing. There is significant cost to these tests. They go on sale around holidays, but even on sale, they're 30, 40, $50. And the Y testing and is particularly much more expensive in the $100 range. So the next Vicky asks, am I correct that only males can take the Y test to trace the paternal lines? That's correct because you do not have a Y, but it doesn't. But it can be anyone along that line. So uh, um, a uncle um, uh, that it comes from your paternal grandfather's line or other people along that line that all have that same an unbroken male line along the one you want to test will work as well. What factors inform which platform to use for Y or MT testing? There's only one platform that does those testing in detail, and that's Family Tree DNA, FTDNA. What are your thoughts about using GEDmatch? So GEDmatch is interesting. First of all, it was one of the first places where you could find people who tested at a different site, um, without having to you yourself test at all those sites. The problem is it's pretty geeky and it's very hard to use the first time, but it has very powerful tools. So what that means is you should be familiar with the other sites, which are much more user-friendly, I think, before you go to GEDmatch. But it is something I do look at, and there are some tools there that are very useful. There's some tools specifically for endogamy. For example, you can also ask the question, is my mother related to my father? One of the things, details I didn't mention is the fact that most of the testing that's done can't, we talked about those two chromosomes, but they can't tell which one they're looking at. And one of the things you can do is if you tested and a parent tested, is you can take your test and the parent's test and figure out which side of the DNA am I looking at? Because remember, there was a paternal side and a maternal side. So you can do what's called phasing. And that's one of the things that you can do at GEDmatch. Some of the other sites do that kind of internally, but you can explicitly do that and create a new kit which has the information that that is specifically says your father's, this is the DNA that came from your father's line, this from your mother's line. If we tested at Ancestry, FTDNA, and 23andMe, is there any value in moving ancestry data to another site? So you don't, the Ancestry test and the FTDNA test and the 23andMe tests are a little different. 23andMe created their own custom chips at one point, but they pretty much get the same results for this kind of work. The one site you didn't say was MyHeritage, and there are some other sites that are used in Europe and other places. I would definitely move one of those to MyHeritage, but and it doesn't matter which one you move.
Dr. Fagan, we have time for one more question.
Yeah. I think there's quite a few queued up here. All right.
Yeah.
Okay. Bill asks, what about genetic testing for genetic diseases? Many our family have carriers, etc. So one of the things that's important is these are not really medical grade testing. Now, 23andMe has BRCA testing for breast cancer and other tests, but they may be testing for one possibility, whereas the formal medical test may be looking for 200 possibilities. So you really need to go with this, but if you find something in here, particularly Ancestry has a medical version, 23andMe has their medical tests. If you find something there, you definitely need to go to the formal medical one, which tests it at a much higher level of detail. So I'm going to stop there and thank you for your attention and listening to the lecture. And I hope you'll be interested in the courses that are coming up.
Thank you, Dr. Fagan. I'm sorry that we can't get to more questions. We do have a tight schedule for today. I'd like to thank you very much for this informative presentation, and we appreciate that you joined us today and clearly for all the time and effort and preparation which went into this. So thank you. If you could see by the comments in the chat, I think everybody is very appreciative. I'd like to also thank everybody from around the world who has participated in this webinar. Please be on the lookout for a follow-up email with a link to the recording and a brief survey. Also, please note that we are planning the full schedule for the fall lineup of Jewish Gen Talks webinars, and we hope to share that information, the entire schedule and list of speakers and topics later in August. So I thank everybody again for joining us. For everybody who's fasting today because of the 17th of Tammuz, I wish you an easy and meaningful fast, and I wish everybody a great day. Thank you so much for joining us.